对，好像导播他是说，可能讲完之后就放2个椅子，然后就跟他对谈，我可能就是会一些容易的一些问题，要诱导他去发挥，就是看他对数学背景的同学有什么，或者在解剖的方面有一些经验。
现在帮我练手，然后接下来我来上课。
好。 好，那我们准时开始吗？都 ready 了吗？
对，你开始他们开始记。
好。Good afternoon, ladies and gentlemen. Welcome to the lecture today. It is a great pleasure and honor for me to introduce the distinguished speaker today, Professor Jonathan Dushoff of Department of Biology at McMaster University, Canada. Professor Dushoff is a famous theological biologist. He specialized in mathematical modeling of infectious disease. He had extensive experience in modeling a variety of infectious agents, including HIV and a lot of other important human pathogens. Let's welcome Professor Dushoff.
I have to use this one and this one, so my hands are full. Good afternoon, thank you for coming. So my name is Jonathan, and I was told as math students and I'm supposed to help you feel interested in modeling biology, so that's kind of my goal. I was also a math student. Now I work on disease modeling and linking between models and data, and I've become very interested in statistics and in the philosophy of statistics, not just doing statistics, but how we think about statistics. And some of the diseases I work on are canine rabies, HIV, influenza, and for the last 2 or 3 years, mostly the COVID-19. I've been mostly unable to avoid that. I'm at McMaster University, as Dr. Fang has said. And I'm also a big promoter of something called the International Clinics on Infectious Disease Dynamics and Data, which is centered in Africa, but we try to train people around the world to do these things that I'm saying, which is bridging between disease models and disease data. 而且有人说，听说我很会讲中文，我其实不会。我中文日常的中文还可以，像请问往板桥265站牌在哪里啊？可是，比方说今天我问一个同事，那个现代的微波炉很复杂，我问他帮我解释怎么用，而且我一点也不懂啊。所以我中文有很基本的，我会试着用一些句话或是写一些句话帮你们。But the basic answer to this question, sorry for those in the audience or in the virtual audience who don't speak English, who don't speak Chinese, the question is, do I speak Chinese? And the answer is not really. And I may throw in some Chinese, especially at the beginning, to try to engage the audience. But if you're an English speaker, I think I don't make any promise, but as an English speaker, you have a better chance of following the lecture compared to a Chinese speaker. And maybe my lecture isn't clear and nobody follows it either way. So I'm going to introduce myself just a little. 我是美国宾州非常人啊，这宾州这非常，而且我大学毕业，我搬到南非洲的Eswatini教书，教2年的书，后来搬回去美国，这不是美国，搬回去美国，在那边认识一个鹿港人，后来搬到台湾，后来结婚住台湾4年左右。那是中华民国85年左右，那时候我们没有手机，所以我那个日常的国语，我学习的日常的国语是很需要，因为手机不可以帮我来这里或怎样啊，都要一直问人家，麻烦问人家。我们再回去美国，我再搬到加拿大，这是我加拿大的城市。所以我心理上都是数学人。而且我怎么从数学到那个怎么说infectious disease modeling， 我今天早上学习已经很紧张，而且我真的第一是真的紧张，第二是真的数学人。我今天早上睡不着，我太紧张了，所以我当下一直想很好玩的数学问题，就觉得很轻松。So this is a picture I made based on the theorem of Pythagoras. We just graph them in the complex plane and you can make art. Here's another very artistic picture that I made based on a geometry question that was asked me by a friend, sorry. And this one is a very interesting picture I made more recently. It's also what I was thinking about this morning. 问题就是棒球的，对不对？你的 three-digit batting average 怎么得到？比方说 if you want a batting average of 333 333，你可以试3次，试3次，按打1次就333。可是你如果要332或是334，你们数学人可以试着 calculate 需要多少的机会才可以到332，蛮多的。So that's how my brain works. And the reason I came to do dynamical modeling is, as you know, I'm also the kind of person who will move to Southern Africa and teach. I want to use math in a way that I feel is helping people. And so for me, that's dynamical modeling, and particularly dynamical modeling of the spread of infectious disease. So what is dynamical modeling? It's the tool that we use to connect scales. If we think about an infectious disease spreading, what we have are millions of events like this one. This is my daughter Mimi. She's like 2 years old, and she's touching this child Nicholas, and Nicholas's nose is kind of wet, and so is she going to get sick? We don't know. We have millions and millions of events like this that add up to very interesting patterns like this one. So these are 1 million or so case reports of measles from England and Wales over many decades. And the question is, what's the connection between what people are doing and these patterns? And can we use these patterns to learn more about what people are doing? So we start with rules, and the rules are based on individuals, and we try to find out how rules link to results, and that's dynamical modeling. And so I think of dynamical modeling as a bridge because it goes both ways. We might study how is HIV spreading in Africa, and what does that tell us about sexual behavior patterns. So that's a way we go from result to rule, but we might also go from rule to result. If we hand out more condoms, or if we close down the Taipei public schools because of COVID, what do we expect the result to be? And I'm not going to talk to you too much about this because I know that most people who are watching this have already heard from Fang Lao Shi and already heard from Ling Lao Shi, and I watched part of their lectures. It's very hard for me because mostly in Chinese, but I feel they covered a lot of what I say about the basics of dynamical modeling. And I can also say you can contact Fang Lao Shi. I also gave a talk about disease modeling at the公卫学院about a month ago. So instead, I'm going to try to talk about some more specific mathematical topics that I think are going to be interesting. But first, I just very briefly review the idea of the normal way of dynamical modeling of the spread of disease, which is that we divide people into compartments. These are people who didn't get sick yet but might. These are people who are sick and infectious, and the blue arrow means they might bring these people over here, and then these people have recovered. And I like to just think about this model as a conceptual model by itself. We don't need any differential equations or discrete time equation or anything to just think about the implications of this model and this blue arrow. But we can implement this in a very standard way, either as differential equations, ODEs, as I'm showing you here, or there's a standard way to implement it as Gillespie equations, as a stochastic model, and it's all very interesting. And then we can ask for different questions about disease spread, what else do we need to add to the model? So we learn a lot already from this model, but we might want to add a delay, right? In real life, I can't get influenza this minute and then give it to you the next minute. It has to reproduce in my body. So we add this delay. This is a model I'm going to talk about later, and you can think about it for a second and try to figure out what's going on. So these people are dead, and so you can say, well, why do these people have a blue arrow? Is because for some diseases, the most important that I know of is Ebola. For some diseases, even after you're dead, you can spread the disease. And so we ask the question, when does this affect our answer about how Ebola is going to spread? Here is a model made by some friends of mine that's about COVID. And they were very interested in making a lot of boxes to try to predict how much medical care was going to be needed during the first wave of COVID in the United States in 2020. So what do we learn from just these simple models? The first thing we learn, and which I know that Lin Lao-Shi and Fang Lao-Shi have talked to you about, is that the disease has the potential to grow exponentially, right? If one person can make 2 people sick, then 2 people can make 4 people sick, and it grows in proportion to the size of the disease population. It has a tendency to oscillate, right? For the measles curves I showed you, they go up and down and up and down, and we know why that is. It's because it's the dynamics of susceptibility. And we have thresholds that some diseases, even if everything is in place for them to spread at a given place in time, if conditions are not favorable enough, they won't spread. And that's the idea of big R, or the basic reproduction number. So that's really what I wanted to review about compartmental models. I hope that's fine for most of you. And then I'm going to talk also more specifically about metrics, how we measure these 2 things. One is how well the disease spreads from person to person as big R, and the other is the speed of exponential growth. So I like to think of big R as the strength of an epidemic. How hard is it going to be to stop? If we don't do anything, how many new cases do we get for each case? And so we define big R as the expected average number of people infected by an infectious individual. And we can think about this in different ways. A way that I really like is as the product of contacts and durations. So that if I have, or if Eric sitting in the back has influenza for 4 days, then we want to know how many people will he contact each day, and what proportion of those people are going to get influenza, and we're going to multiply that by 4 days. And if we do it right, we get a unitless quantity. All of the units cancel out, and we have the number of new cases expected per case. And as soon, I'm sorry, I'm having trouble with the buttons and with the microphone. It's all very exciting. As soon as we think about it this way, then we realize that there's a threshold that's not 0, right? If we have a disease in a place, but each case causes less than 1 new case, the disease 会自己不见了，自己越来越少。So this was a big idea of over 100 years ago, first promoted by Ronald Ross. He said that we can do a simple calculation and say that the relationship between, say, the effective number of mosquitoes and the expected level of yellow fever follows a shape like this. So if we go here, if we think that our system is here and we reduce the number of mosquitoes by 80%, we don't reduce the incidence of yellow fever by 80%, we reduce the incidence of yellow fever to 0. And the idea of big R is very helpful for thinking about why diseases come and go in some places. In other places, I gave the example here on the slide of dengue in Taiwan, because we worry a lot about dengue in Kaohsiung and Pingtung, and we don't worry a lot about dengue in Taipei or Taoyuan. Why not? Taipei, Taoyuan have the same, not the same, but they have mosquitoes, and they have cases of dengue coming from all over Asia, and dengue can spread here just fine, but as long as the R0 is less than 1, as long as the average number of cases per case is less than 1, we still watch it, but we don't need to worry about it the way we need to do in the South. And so we mentioned the tendency of exponential growth, and I'm just going to attach this to a number which we call little r, and since you're all math people, I'll just say that r has, I don't know if you're all math people, since I prepared this talk for math people, I'll just say that r, little r, unlike big R, has units, and we think about it as a rate of exponential growth. And just like the stronger the disease is, the harder we expect it to be to control in certain ways. The faster the disease is, the harder it is to control, and certainly the less time we have to control it. And so we're very interested in little r, or the reciprocal of little r, which I like to call the characteristic time, and which is closely related to the doubling time, which was a very popular metric back when we were also worried about COVID. So here's an example of an exponentially growing disease. HIV grew exponentially in Africa for many years, and it was very scary, and people were projecting this exponential curve and forecasting an even worse disaster than the one that came. So the main thing I want to talk to you about today is what I call transmission intervals or generation intervals, the amount of time between when Eric gets sick and when I get sick because he infected me, or the amount of time between when Eric gets reported to the CDC and when I get reported to the CDC. We have lots of different ways of thinking of transmission intervals, but I'm going to associate each of them with a single disease generation. So Eric gets me sick, and apologies, but I infect Dr. Fang, and each of those is a disease generation. We want to know how long it takes. And these transmission intervals used to be very unpopular. So this is a picture of some rich people who are trying not to pay attention to their poor relatives. And it reminds me of the way mathematicians used to think about transmission intervals. We like to solve equations and fit curves, but we don't want to think too much about how long it takes for a person to pass disease to another person. COVID changed that to a large extent, but people are still often dealing with generation intervals using ad hoc methods. And I found, starting with the Ebola outbreak about 10 years ago, that a lot of smart people didn't understand what they should about transmission intervals, and it's been one of the main focuses of my work ever since, and it's kind of fun math, or I think it is, so we're going to talk about it for a while. So here's a picture. I think most of you got it when I said it, but a generation interval. This girl gets sick on day 3, and she infects this girl. Actually, she gets infected on day 3, and she infects this girl on day 8. So that's sometimes called, the names are very confusing, and I'm sorry about this, but this one from transmission to transmission is usually called the generation interval, even though in some sense they're all generation intervals, and maybe this one should be called a transmission interval. And here's some work some colleagues of mine did during the Ebola outbreak, or this is actually a picture I made based on their work, and this is an estimate of an Ebola transmission interval distribution. So if I get infected with Ebola, my chance of passing it to somebody in the first 5 days is extremely low, and then it's higher, and then it's higher. And we try to estimate relationships like this and use them to understand the other parameters of the disease, and in particular to understand little r and big R. And in fact, transmission intervals are the link between little r and big R. At any point in time, we have people getting infected, and if each of them is causing a certain number of new infections over a time distribution, that's what causes the exponential growth of the disease. And we can ask, for example, as we did in Ebola, what happens if our generation estimate changes? So let's say we think the distribution looks like this, and then somebody brings to our mind, you remember I showed you the picture of the dead people transmitting, people say no. We think dead people are transmitting more than we thought. So how much Ebola spread is occurring after death? This is a picture after people started having this concern. They established these safe burial teams. These are people who are trained both culturally and biologically. So they go and they help you have a funeral that respects the dead person according to your culture, but also does not infect anybody else in your family with Ebola. And specifically, and this, I guess, is one of the principles of public health, specifically can't infect one of the members of the team with Ebola. And so they're particularly well protected, and they're the ones who interact with the dead person. So we don't know. There are many different cultures. The Ebola was spreading in 3 different countries. But we asked the question, if we find out that Ebola is spreading slower than we thought, is that a good thing or is that a bad thing? So in other words, if the generation interval, if the amount of time I spend spreading Ebola is slower than I thought, if there's more after death transmission and maybe, well, let's just stop there. So do we think that's a good thing or a bad thing? The goal here is to get an answer. I even bothered to write all this in Chinese. I have no idea what it says. 好，有几个人觉得发现比较慢是好的，空间比较多，对不对？所以你没有错。还有没有人觉得不好？因为比较有机会。对 ，Okay, so 2 to 1. The answer is 不一定。The answer is 要看情况。And it depends in a very useful way, because what it depends is on what we think we know. So as a mathematician and the tools I learned at university and in graduate school, we tend to start from parameters. So if big R is known, if we know each person is going to infect 2 new people, then making the generation interval slower is good news, right? Each person's infecting 2 people. Now it takes them longer. That's good news. And so we can say quicker generations leads to faster population speed. The opposite of that is that slower generations, if they have burial transmission and the parameters are staying the same, then the disease spreads more slowly. Okay? So that's not a wrong answer, but that's not the case. That's not what happened with Ebola here, because we don't know the parameters. Very often, so I was just checking to see if I wrote Chinese at the top of that slide. I don't know if it's useful. 如果知道那个 rules or parameters, then slow transmission is good news. But what we knew was the result, not the parameters. What we knew, these are real data from the Ebola epidemic when we were having this conversation. What we knew was little r. And this value of little r is consistent with many different possible parameters. So we don't know what big R is at all, and in fact the data can't tell us. The data only tell us little r, and if little r is what we know, then slow spread is bad news. And why is that? This is a great picture of the idea. If little r is what we know, what it means is we know it takes 2 weeks, in this case, to get to 16 cases, okay? So if the generations are fast, what that means is that each generation needs to multiply by 2 to reach this destination. But if the generations are slower than we thought, then what we need is more strength. We need each person causing more cases. And so, in fact, we get an opposite answer to the question of is slower transmission good or bad, depending on whether we think we know little r or big R. And this was really, I think, the trigger for me to really get into generation intervals, because people were showing this at the time, but they were showing it with very complicated models. They would make many assumptions and estimate some parameters, and they would say, okay, in this case, we can see that more burial transmission means higher big R, but there was not this general idea at the time that we just understand this concept. And one way to think about it, these 2 opposite answers are actually closely related, because we can think of the population level spread, and we discussed this a little bit when I asked you to raise your hand. It results directly from these 2 things. How fast does each person spread it, and how much does each person spread it? And so we can think of it like we're multiplying. There's something about the strength, and we're multiplying it by something about the quickness, something about how quickly each individual does it. And I have to use the microphone or not? I find the microphone very confusing. I should use it, okay, because I'm always, okay. It's a good thing I'm not a pop star. So if this is a product, if little r is somehow like big R times the generation speed, then it makes perfect sense that when we change the generation speed, if we know big R, that means we're going to get a bigger little r. And if we know little r, then we're going to be dividing and we're going to get a smaller big R. And so this result, which is often surprising to people, I didn't try hard enough maybe to trick you because I wanted you to feel included. Often you can get people to say, no, faster generations should be more dangerous. It's a surprising result, but it actually comes straight out of the same framework. So what is a generation interval? In terms of the modeling framework that we introduced with Professor Lin and Professor Fang, we often think of the generation interval as a latent period. How long does it take me to move from box E to box I and become infectious, plus what I call an infectious waiting time. And those of you who have studied these differential equation models may be aware that we think of the infectious waiting time as having the same distribution as the infectious period. And this is something which confuses people over and over again, and which I thought would be fun, not funny, I hope, fun to talk to about in a math term. And I like to think about this problem. So let's imagine that we're waiting for a bus, and we know the bus leaves the station every 10 minutes on average, and we're going to assume that the time for the bus to leave the station is an exponential distribution. It has no memory, so they're leaving independently. I wrote that in Chinese, but no, I'm too shy to say it. How long do you expect to wait at the bus stop if the buses are leaving the station every 10 minutes independently?可以用中文啊。如果慢说，应该要等多久？每10分钟有一个公车，大概要等多久？哦，所以范老师说5分钟，有没有人不同意？哈？ At most 10 minutes, even if it's an exponential distribution. Interesting. Okay, that's not what I expect. So, 5分钟 is the right answer to the wrong question. If the buses leave every 10 minutes exactly, John, sure, then you wait 5 minutes. But we all know that they don't, right? We know if there's a bus on average every 10 minutes, then we expect to wait more than 10 minutes. Last week I went to the bus station across from Gongwei Yuan, and there were 4 buses at the station, and I missed them all. I was about half a block away. And by the time I got to the station from half a block away, there was another bus, all 265. So no, we expect to wait more than 5 minutes. If it's an exponential distribution, does anybody know the answer to the official question? Okay, so if the bus is, maybe I'll try to put it in a different term. So when we say 彼此独立,意思是 at any, let's say, 6 seconds, there's 1% probability a bus will leave the station, but it doesn't care. It doesn't care if the last bus left a minute ago or an hour ago or a year ago. We still have that same fixed probability. Okay, so if that's the case, if the buses don't care about each other, on average, how long do you expect to wait? Okay, so this is interesting, and I'm sorry that I got stuck on it. the answer is 10 minutes, right? If the bus doesn't care how long it's been, then no matter when I get there, no matter when I get to the station or to the stop, on average I would expect to wait 10 minutes because it doesn't depend on the history of what I've done. And that's exactly how the simple disease models work, and that's why the infectious waiting period, the average waiting time until somebody infects somebody, can be the same as the infectious period. Is there any way I can make this clear to people, or can anybody ask a question? Do people believe me that if the buses are really independent, I would wait 10 minutes on average? Okay, so let's just believe it for a minute, and it's on tape, so maybe we can go back and study it, although people are also free to ask questions at any time. So what's interesting here, when I get to the station, I'm going to wait for 10 minutes. But if I look backwards in time, and I ask when was the last bus come to the station, it's also 10 minutes ago. So the interval between buses at the station is 20 minutes, even though, at the bus stop is 20 minutes, even though we assume that it's 10 minutes. So how is that possible? The answer is that we're averaging different things. 10 minutes is the average amount of time from the point of view of a bus, of an interval between buses. But if instead we pick a time at random, we're more likely to be in a long interval than in a short interval, right? The 5 buses came in a row. Very few people caught the third or fourth of those 5 buses. But a lot of people came after the 5th bus, and they had to wait another 10 minutes. So the average depends on how we're averaging. And it's exactly the same as transmission. If some people are infectious for longer, if we take the average over people of how long they're infectious, we're going to get a different answer than if we take the average over transmissions. People who are infectious for longer are more likely to make others sick. And it's exactly the same, just as with the buses. If there's an exponential distribution, the average gap between buses based on time is twice the average based on buses. But as Fanlauscher already said, we only expect to wait half of that time. So it just happens to balance out. If the distribution is exponential, then the amount of time I wait at the stop, the time from my point of view, is the same as the time from the point of view of the buses or the person who controls the buses. If the distribution's not exponential, it's not the same. If the buses are perfectly joined, sure, then the amount of time I wait is less than the amount of time between buses. And if the buses are doing something crazy, then maybe the time I wait is going to be more. And there's some math about this, which maybe I can say quickly, but I feel like maybe I should pass on. The time until the bus comes, on average, is equal to the average time between buses, multiplied by a factor that reflects this variation, but then divided by 2, because on average, you're going to come somewhere in the middle of this interval. Right, so, sorry. The Chinese is constantly confusing to me when I'm nervous. When I was writing it yesterday, it all made perfect sense. Anyway, these are just the Chinese labels for the different kinds of transmission intervals. So the main transmission interval, the one we call the generation interval, is from transmission to transmission. It's from when I infect you till when you infect your friend. We can also, and these are the things that actually are the mechanism for the epidemic moving forward, right? This is when the disease gets spread, but we usually can't see it. We don't know exactly when we got infected with COVID. We certainly don't know exactly when we got infected with Gamao. So often we study other intervals that we can have observations. Classically, the when you start feeling sick is called the serial interval. But when I study real disease data, there's all kinds of different data streams that may be more reliable. So often we look at when did the doctor decide you were sick, or when did the doctor tell the CDC you were sick, and we're going to come later in the talk to talk about how these intervals relate to each other, because there's also interesting math there. We can also mention that with COVID, we talk a lot about serial intervals, but it's possible to pass COVID on to someone else without ever really feeling symptoms. And then how do we even say, what does the serial interval mean in a case where one of the people in the chain didn't have any symptoms? So we're trying to measure these intervals. And the other things besides Ebola that got me started on this was actually a theoretical exploration, which is that people who model simple diseases, disease systems, noticed that these generation intervals weren't what they expected. And we were able to unravel this to some extent. Others had done it before us. But if you look at generation intervals ending at a particular time, for example, at the beginning of an epidemic, when numbers are going up, they're going to look short. So if we're looking at somebody who got infected with HIV in Africa in 1994, the person who infected them got infected probably 2 or 3 years ago, not just because that's how fast HIV is, but because 5 years ago there was no HIV in their country. So when the epidemic is growing quickly, there's a bias. Most of the people who could infect you were infected recently. And this is a bias that can carry over for a long time. A lot of people are infectious right now, and whenever we stop, not whenever we stop looking, but if we stop looking near the beginning of an epidemic, the measured generation intervals are going to be shorter than what we call intrinsic, shorter than the profile we would get if we look at a sick individual. So we're biased in many cases towards observing faster events, and we defined this intrinsic interval, how infectious am I, a forward interval, if we take a cohort of people, if we say who got infected with COVID today and when will they infect others, or a backwards interval, a COVID of people who got infected with COVID today and who infected them. And we can explore the different biases that affect these things. I told you the bias that affects the backward interval. The forward interval is less biased, but it may be fun to think about what would affect it. And I guess the question is, what other than intrinsic infectiousness would change my ability to infect people through time? And then we can also just ask the question from a practical point of view, if we're at the beginning of an outbreak, what do the intervals that we're able to observe up until now look like? And the answer is, it's a weighted average of backward intervals. And if we understand the biases in backward intervals, we can try to correct and use the data that we have. And we were able to make a simple model and show that this just works. So here's a simulation model done by a student of mine, and we look backwards in time. We take these cohorts and we say, at a given date, what was the average backward cohort interval, and we calculate it based on this exponential bias, and we show that the theory matches the simulation, and this is now something people use. It's very complicated in the real world, but it's something that people use to try to understand transmission interval measurement in real time. So I had said before that one of my main interests in generation intervals is the way that they link little r and big R, and we talked about the example of the Ebola. So there's definitely something missing or out of order from my talk. Okay, that's not good at all. Okay, so I'm supposed to have some slides about what I mean by K of tau, and maybe they're going to come up later. Now I want to cry. Intrinsic. Don't know. How do I make it big again? So K of tau, I'm just going to ask you to imagine. Tau is the amount of time since I got infected with a disease, and K is the expected intensity of transmission. So we think of K as a transmission kernel, and we ask the question, how does K relate to little r and big R? And the answer is from hundreds of years ago, the Euler-Lotka equation, and it's very beautiful. You find the value of little r that if you assume the disease was growing exponentially at that rate, that the discounted contributions add up to 1. So in other words, if the total number of people you expect me to infect is bigger than 1, then we're going to figure out at what rate does the disease population need to grow exponentially to balance that out. Or if the total value of K integrated out to less than 1, meaning that I would be expected to infect less than 1 person, then the exponential rate of growth is going to be negative, and we're going to inflate these infections and again balance out to 1. And so we can solve this equation for little r, or we can invert it. We can invert it, and we write the generation interval as little r, as big R. Sorry, we write K as the product of big R and the generation interval distribution, and we get this mathematical relationship. Okay. I've been talking, I guess, for almost my 45 minutes, and I really don't know what happened to that equation, and I'm not sure what to do about it. But we can take this kernel, and we can factor it in 2 different ways that help us think about the relationship between the kernel and the strength and the speed of the epidemic. So if we think about big R as being the area under a transmission kernel, then we can factor the transmission kernel into a distribution, which is the generation interval, and a multiplier, which is big R, the expected number of cases. If I factor out the total, then this becomes a distribution. Or if I think about it from the Euler-Lotka point of view, I can say, well, how quickly? So in this case, I could say, what proportion of the population would I need to vaccinate to bring this transmission kernel down to where it only adds up to 1, where the disease is going to be balanced? And in this case, I can say, how quickly do I need to find people? If I'm tracing Ebola cases, and I'm always trying to find people who might be in danger, and I find them and isolate them, here I'm saying, how quickly do I need to find them? And what we did is we tried to explain these 2 as alternative interpretations of how hard it is to control a disease, whether we use a strength perspective or a speed perspective. And I am now, it's been about 40 minutes, and I'm mostly confused because my equations disappeared. But I'm wondering what else I should try to tell you. So I'm going to skip this part because it doesn't have the equations and because I'm running out of time. And I'll talk just a little bit more about another fun math piece, which is other distributions. So we talked about the transmission interval, the time from when I'm infected to when I infect you is the thing we want. but we usually can't observe it. We're usually observing things that we tend to loosely call serial interval distributions, the distribution between when I get sick and when you get sick, or between when I get reported and when you get reported. Here's a picture we made when studying rabies of serial interval distribution. So we imagine this is the first, so rabies, you probably know, and it's probably going to appear in Chinese later, but it's a disease mostly of domestic dogs. In a lot of parts of Africa and Southeastern Asia, it's still a very serious disease that spills over to people and kills a substantial number of people. And here we're imagining a dog gets bitten, and this dog eventually develops symptoms, bites another dog, and this other dog eventually develops symptoms. And so in this case, we can measure both the generation interval and the serial interval. The one is from bite to bite, and the other is from illness to illness. So I think I figured out what happened to my equations. And we usually expect, well, we would expect the serial interval. We measure the serial interval, think that it's the same as the generation interval, right? They're the generations of the same process. But in fact, these distributions are not the same. And this has been known for a long time, that the serial interval usually has more variation than the generation interval. And the classic example of this is influenza, because in influenza, it's not rare for serial intervals to be negative. So I can get influenza, this is perfectly realistic. I could get influenza on Sunday, I could infect you on Tuesday, and you might go to the doctor feeling sick on Thursday, and I might go to the doctor feeling sick on Friday, right? So I'm reported on Friday, and you're reported on Thursday, but I infected you. The serial interval is negative, it's backwards, okay? That cannot happen with the generation interval. If I get infected on Tuesday, I can't infect you on Monday. So the serial interval is more variable than the generation interval and can even be negative, and this has been known for quite a while, but it also raises a very interesting problem. We do expect them to have the same mean, though, because they're generations of the same process. Remember, Eric's infecting me, and I'm infecting Professor Fang. We haven't decided who Professor Fang is infecting yet. But it seems like the times have to average out. But when we did this study for rabies, which is a really good disease because people live with these dogs and can observe them getting bitten and can observe them getting sick, we did not find that. We observe the generation interval directly, we compare it with the serial interval, and we find that the serial interval is longer. In other words, the time between transmission and transmission on average is longer than the time between symptom and symptom. And how is that possible? It's possible because the disease is acting differently on average in different dogs, and because the generation interval is measuring how long it takes a dog to get sick plus how long it takes that dog to then bite somebody else, only if that dog bites somebody else, right? If I don't bite anybody, I'm a dog now. If I don't bite anybody, I don't have a generation interval. And if I bite 10 other dogs, I have 10 generation intervals. And that always has to be true for the waiting period, right? The waiting period, how long it takes me to bite you, only makes sense if I bite you. But in fact, in rabies, the latent period is much longer and much more important. And for the serial interval, we see the serial interval in every dog that's bitten, whether they're bitten or not. So if there's some correlation, if there's differences between dogs or differences between the course of disease that leads to a correlation between how long is the latent period and how much I bite, then we can get these differences. So this is cool, and this was something that people hadn't really thought about until we discovered it by accident, and is relevant to other diseases. So people who are diagnosed early, who have a shorter time to diagnosis, are also less likely to transmit. And that's going to lead to a systematic difference between the serial interval and the generation interval. And I'm going to end with one more thing that's very hard to understand, and which I think is mathematically really cool, which is this idea that I had for years, and many other people had for years, that the serial interval has the same mean but a different variation as the generation interval, turns out to be impossible. Even if we assume that everybody's the same, in other words, right, we assume that everybody's the same, and we think about, instead of a process where infections cause infections, we think about a process where symptoms cause symptoms. And we solve the same equation, the Euler-Lotka equation, and I'll apologize one more time for messing up my slides for that equation. But the serial interval distribution has to give the same link between little r and big R as the true process, and that's impossible, because if the generation interval is wider, then that's the same as spreading faster. The wider my variation is, if the two distributions have the same mean but a different variation, then the one with a higher variation is going to spread more, right? Because I might, by chance, infect you in 2 days, and then you're going to infect somebody else in 2 days, and so forth. And so what that means is that if they really had the same mean and a different variance, that the serial interval would be predicting a lower big R for the same small r, or a higher small r for the same big R. But that's impossible, because they're just two different ways of describing the same disease spreading through the same population. And the answer is kind of cool. The answer is the same kind of bias that we found when we were talking about generation intervals early in an epidemic, which is that early in the epidemic, if I look at forward intervals, I would expect them not to be changing, right? The population is growing exponentially, everything is fine, and that's really true of the forward generation interval. But if we're thinking mechanistically, if we're thinking about what actually happens, a forward serial interval has a backward component. We're asking, when did I get sick and when did Eric get sick? And so to do that calculation, in some sense, Eric had to be infected before he infected me. And early in the epidemic, if things are increasing quickly, there's a bias, right? There were more people who had COVID 2 days ago than 5 days ago. And so Eric is more likely to have got infected more recently. And that means that the forward serial intervals that matter are actually longer. And that's what balances, longer on average. And that's what balances out this inconsistency. And I was very sad, because I was confused for a decade, and then I found this paradox and I was so excited about it, and then one of my students found the answer before I did, and I'm still jealous of him for finding the answer before I did. But it works perfectly. So these are simulations again, and we can see that early in the epidemic, things are growing exponentially, and so none of these periods change. Here we're looking forward or backward at the incubation period, the generation interval, and the serial interval. But what we see is that early in the epidemic, the backward incubation periods are short, The forward incubation periods almost never change at all. And the result of that is that early in the epidemic, where we had this inconsistency, the average serial interval, even when everybody's the same, the average serial interval is still not the same length as the average generation interval. So that's most of what I wanted to say. My summary, I like thinking about things in terms of capital R and small r, and I'm trying to challenge. There's this idea that many of us learned that big R is the important thing that we use to understand how the epidemic is controlled. I feel that speed is actually complementary to big R, and that this factorization shows that there's just two ways of looking at things. Transmission intervals are key, and they're complicated. We need clear definitions of the different kinds of transmission intervals we're looking at if we want to be able to understand them. And then I would just say that if you love math and you want to do something useful, you should think about dynamical modeling and/or dynamical statistics, or talk to me, or find out about ICI 3D. And thank you for your attention and for the invitation.
OK， 好。Professor Dushoff, thank you very much for your very refreshing lecture on exploring the fascinating mathematical property of an important biological quantity, the series interval. And you fully apply the mathematical tool in analyzing this biological phenomenon. So could you explain a little more about the field of serological biology? Because I think this field is relatively underdeveloped in Taiwan, particularly among the mathematical community.
So that's it. Hello?
This. Okay.
Me. Thank you for the question. I'm sure you know much more about it than I do. I don't always see it as completely separate, right? I see myself as somebody who does science. You know, I'm in a biology department.
Yes.
But really, I try to find scientific questions that can be answered with mathematical tools, and I think people can do that in many different areas, right? I mean, we've talked about how some problems are fundamentally dynamical problems. If you're trying to look at how things are linked across scales, you can't do that without dynamics. A thing that I haven't talked about much is statistics, and of course, we've all learned some of the old-fashioned statistical rules in machines, but I think that thinking about statistics from sort of a theoretical perspective and developing your own statistical philosophy can also be a very productive way forward. In terms of how to get universities or funders to support it, I don't really know. I certainly don't know about Taiwan. I've been very lucky, but I don't know why.
Could you share your experience from a scholar in the with a mathematical background, and how to you study the biological phenomenon? Because I think many mathematical students in Taiwan find biology somehow very difficult, and a lot of Latin name or a lot of things to memorize.
Yeah, so there's a lot of interesting aspects, I think, to that question. I did learn a lot of biology,
Yes.
but I mostly didn't learn it as an undergraduate. And it's when you're talking about the memorizing the Latin names, it's very right. A lot of teaching, certainly a lot of teaching in my country, Canada, is kind of stupid. The professor, it's important to the professor to give you a grade and to spread the students out by grades. My daughter is now 我们说大一嘛, first year undergraduate, and she's learning such interesting things, but she doesn't have time to learn because she has to study long lists of stupidity for the test. And biology can be like that. We need to find ways to train people in biology, to train people with less memorization. When I was an undergraduate, I kind of ran away from the biology because it was kind of stupid. Stupid not in the sense of stupid, but stupid in the sense of the way it's tested, that you have to spend so much time learning things. And I can tell even a story. I took one biochemistry class, and they said, this is my first year in university, they said, learn all the amino acids. And so I just assumed they mean I should know their names and have some idea what they do, how they group. And no, on the first test, they asked us to just draw all 20 amino acids. And I said, that's stupid. Why would I learn that? And that was even before we had the internet. It's just become more stupid. I don't know what the roots are. I think if you can learning the basic ideas of biology is important. If how you can find a way to do that and get credit for it without getting a headache and learning a whole bunch of crazy stuff, I don't know. As I say, for me, I was lucky. I learned some basics in undergraduate, and then I came back to it in graduate school where people were much less focused on making me memorize things. You should not be memorizing Latin names. You should not be memorizing anatomy charts. You should not be memorizing the details of huge chemical structures. But I don't know what the answer is to what you should do to get some of that knowledge.
Thank you for sharing your perspective. I think your word will encourage our student in the Department of Mathematics. Actually, it's not necessary to memorize a lot of nonsense. What important is to understand. So you are a very good role model, I think, for the next generation student. Biology is understandable, not necessary a task to memorize.
Yeah, I mean, biology is in fact very hard, and it also depends what level. I've recently done more population biology.
Yes.
In some sense, because I understand it better. When we start looking at things like immune system or biochemical pathways, it really is more difficult. So it's not that it's easy, but people make it even harder than it should be, or people teach you not the parts that you need to understand.
Can you share the first topic your research in the early stage in your career? I think it will be a lesson for all of us. So how do you identify an interesting topic to investigate?
Okay. So those are different questions. So I started, when I started doing mathematical biology was a long time ago, and not as much was known. And I was very lucky. I was able to do some simple mathematical calculations about R0 and about population mixing that even then, and I think this is a lot of what I do too, even then had already been done. It was already kind of known, but it hadn't been explained carefully and clearly in a way that maybe could be applied better. And it's still what I do, is I try to understand things, and then because I try to ask a scientific question that has a mathematical, dynamical, or statistic component. I try to figure out some of the basic principles, and I often try to explain them to others if I feel that I can figure them out for myself, even though I'm very often not the first person to figure it out. And I don't know that that's always bad. Sometimes things need to be explained more than once before they're understood. A famous version of that is, I talked today about the Gillespie algorithm. Do people know the Gillespie algorithm? Okay, the Gillespie algorithm is when I said there's a standard way to implement a simple model as ordinary differential equations, and there's also a standard way to implement it as an individual-based model. So instead of saying that we have one person with flu today and 1.1 tomorrow and 1.21 the next day, we can say, well, if we have one today, then tomorrow we're either going to have 2 or we're going to have 0. And there's a standard, simple, beautiful way to do that based really on the same assumptions that you use for your differential equations. The only additional assumption that you need is that individuals are to be treated as individuals. And that's called the Gillespie algorithm. And the reason I bring that all up is Gillespie did not invent the Gillespie algorithm. The Gillespie algorithm was perfectly well known among mathematical disease modelers long before Gillespie. But Gillespie explained it in a way that people in a much broader area understood it and were able to use it. And one of my professors said that the thing is not named after the first person who finds it. The thing is named after the last person who finds it, right? And so part of the story is finding it, and part of the story is being able to explain it to others or to be able to apply it in ways that people can use it.
Oh, very, very interesting. And could you share your experience studying HIV in South Africa? You just said that you had stay and work in South Africa for about several years.
So that's an interesting question.
Yes.
And I've never, in some sense, the short answer is that I've never really done what I think you're picturing. So I was in Swaziland when the HIV epidemic started, but nobody knew. Nobody knew. And then I started in 2007 teaching every year in South Africa and studying HIV, but I was never, a couple of times I consulted people, I was never directly involved in an HIV study. In collecting HIV data. Yes. So the HIV work I did, some of which is interesting, was really about using the data that was already there. The main thing that I remember finding interesting is we used survey data from something it's now called Measure DHS for demographic and health surveys, and they talked to people about their sexual history and collected blood from them to do HIV tests. And we were able to learn about the roots of HIV transmission, meaning the question of how many people were getting HIV. We studied people in long-term partnerships. And so the question is, if you have HIV, did you get it from your partner, or did you get it before you met your partner, or did you get it while you were with your partner but from somebody else? And so that was interesting and had some implications for the policy at the time. And yeah, I'm not sure. I could talk for a while. I'm having trouble remembering the details, but the answer is that I didn't involve myself directly in collecting data, but I was talking to people who were asking interesting questions, and I was finding the data that already existed, and I was thinking about new mathematical or statistical ways to engage with it.
Thank you for pointing out another very important point. Traditionally, mathematicians work by themselves alone. However, when we do infectious disease modeling, actually the data is quite important. So could you highlight a little more important collaboration with other scholar or data source?
Yeah, so this is not, I mean, the good data collaborations has not really been my strength. I've mostly used public data.
Yes.
Certainly, and there's nothing wrong with using public data. A lot of people are spending a lot of time collecting data, and it's nice if it can be used properly. And through the years, I've used data from many different sources. I guess many of you are aware that there's a lot of COVID data available from different governments. I guess one of my best collaborations has been the rabies. So I showed you some rabies data. That's a good friend of mine who's been collecting rabies data for decades in Tanzania, and also goes around the world and consults with practical people. And I guess I'm sort of, as the math person, I sort of consult with the person who consults. You know, I'm this second level of that. But that's been very productive. That's Katie Hampson. She's now at the University of Glasgow, and I met her in Princeton.
Thank you. So, so it is. So thank you for highlight. Actually, there are a lot of public data can be used. So ideally, if we have good collaboration with a colleague in biology or public health, we can get very good, very interesting data. However, there are also plenty of public data available.
Also, I'll say here in Taiwan, wherever I am, the公卫学院 is right across the street from CDC. And I know a lot of people, probably including you, have good collaboration with the CDC. And it can be very good. You know, using public data is good. But when the person who's ready to use the data dynamically is also able to talk to the people collecting the data, that can help a lot, right, in terms of what should be collected. We've had a lot of long discussions during COVID. And during COVID, it was very hard because everybody thought everything was an emergency all the time. But a thing that I spent a lot of time telling COVID people is, if you're going to test a biological sample, you should record why you tested it. And this is a big thing that as a modeler you understand can be very important, but the people who are collecting the data don't understand. So if I'm going to test a sample to say whether it has COVID antibodies, to see to do a genetic sequence, to do a sequence of a marker and see whether it's Omicron or not. How did I choose that? Is it somebody who was sick? Is it a random sample? Is it somebody who I stopped at the border? Just writing that down, because for me, the idea they can do a whole viral sequence and tell me every single every single base of DNA in the virus is a huge job. And the idea to write down, okay, this person was 43 years old and we collected the sample for this reason, is such a tiny job, but it makes the huge job so much more valuable. And the people collecting the data don't know that unless we tell them.
Thank you. And I think mathematical modeling not only can study infectious disease among humans, but also for animal infectious disease, such as mad cow disease. Could you comment a little?
I agree. I mean, we study rabies mostly in the dogs because that's mostly where it spreads. I don't know that I've been involved in any other, no, that can't be right. Rabies spreads among the dogs. We've also studied to some extent how it spreads among other wild animals in Africa, the jackals and lions and so on. Some kinds of malaria can also spread from people to animals and back.
Yes.
And Ebola also spreads. I agree that it's the same sort of techniques and the same sort of intuition, right? And in a sense, a big part of understanding dynamics and a big part of the reason that we like to learn things like the SIR model is that they give us a dynamical intuition. We sort of understand something about this bridge between big events and small events.
So you are also specialized in the statistical analysis on infectious disease data.
I am also very interested in studying statistics and I'm very interested in statistical philosophy.
Yes.
And there's two sides of that. One is just how we should think about and talk about and validate statistical methods. And then the other is what I would call dynamical statistics, right? So if you have a dynamical model,
Yes.
doing statistics, getting confidence intervals is harder than for a regular model. And one of the parts I skipped because of time was talking about how to look at different sources of uncertainty in a dynamical model.
Thank you. Thank you for your sharing experience. I think that most the audience are students in Department of Mathematics, or some students are in Department of Physics. So could you give some encouragement or advice, career advice for the audience?
Well, as I said, I've been lucky. I never feel like I can give people career advice because I don't know why I've been successful or how to help you be successful. I would say that if you're going to be a scholar, if you're going to study, you should do something that you find interesting, right? And maybe you've already thought of that. For me, though, I did something a little different. I mean, what I love the most is the kind of crazy stuff that I saw you that I showed you at the beginning, with the baseball and the circles. And I still do that, like in my free time. But what I decided to do was work on problems that I thought were going to be helpful. And I did that because I wanted to. I did that for the same reason that I went to Africa. But it may also be a good strategy, right? If you can find something you like and is also useful to people, then you may have more chance to find a job or to find a funding if you find the job.
Thank you. Perhaps we can have a few questions from the audience. Any question from the audience? Okay.
Thank you very much. And I wonder some question about the rabies virus. Because the rabies virus is an important zoonotic in human, and most of the infected human always bitten by wild animal or domestic animal. And if we want to build a dynamic model, how we can estimate the parameter between the animal and human? Thank you.
Right. So the big problem with a rabies model is to figure out how much it's spreading in the animals, right? Because that's a dynamic process, and that's going to be the main thing that changes in terms of the risk from animals to humans. So in places where there's a lot of rabies in domestic dogs, you can do a reasonably good job of estimating the risk to a person. What are the factors that translate from the level in dogs to the risk to a person? In other places where there may be much less rabies, right? Like if in Canada we have rabies in raccoons now, but it's very small and they very rarely bite the people, and so it's hard. You said the epiparameters. So by that, do you mean specifically the part of how it gets from the animal to the person? So that, in some sense, can be measured similar to other epiparameters, in my mind. The hard part is knowing what's going to happen in the animals. It's the first hard part. The other hard part is that when the risk is very low, then the uncertainty is going to be very high.
One more question. Yes, Professor Lin.
Okay, thank you for your wonderful talk. You just mentioned, if I didn't make a mistake, you just mentioned immune system. I'm wondering, I mean, you talk about this population biology and infectious disease, and then which is sort of, I think it's good you build up some mathematical modeling and then try to, that might help the prevention or control on the disease spreading, this kind of issue. But I'm wondering if you can introduce also the other direction, like inside our bodies, like immune system, and then how mathematics modeling can help us to understand this immune system.
Right, so that's a great question, and I've been in and out of thinking about the immune system for many decades. And I'll just mention that just like the picture I showed at the beginning, where we have two children touching each other, and what we want to know is how it affects a whole country. The immune system is exactly like that. It's a dynamical problem, right? We have cells interacting with cells and antibodies interacting with viruses. And what we want to know is the result at the higher level. So it's fundamentally a dynamical problem. It seems much harder. Like the immune system just seems to me to be very complicated and very hard to understand. There's been a lot done. I mean, you may know with HIV in particular, there was a lot of, well, for any subject, when you start the dynamical modeling of the immune system, you learn some exciting things at the beginning, and then you seem to get confused because our immune system is just so complicated. And the problem it's solving is so complicated, right? It's all these little cells that are mostly independent, and they have to kill the things that are trying to kill you, but not kill you, which they very much have the capacity to do. So recently, because Canada has some big initiatives now to try to understand disease spread better because of COVID, and I've gotten a little back into immune system modeling. And yeah, it's cool. Some of the principles apply, but I've found it harder. There's also sort of hybrid where we can take these SIR models and be what I would call phenomenological, that we put a little bit of idea of how immune people are going to be at different times and under different conditions without worrying too much about why. And so people have done a lot of models with that about flu. Like if you've had this flu, how badly are you going to be affected with that flu without ever modeling the T cells and the B cells and so forth. Yeah, I'm spend a lot of time wondering how much I should think about the immune system, including recently.
Thank you. Any more question or comment? Yes?
Hi, Professor. Thank you for your talk. And since you are a theorist in dynamic system, and I'm wondering that a kind of field in dynamic system, particularly in the nonlinear dynamic system, there has a study called bifurcations. So does the bifurcation, did you study that, and how is this related to the real problem like COVID model or something? Yeah, can you share some?
Right, that's a great question, and I used to study bifurcations a lot. And it's a whole topic I haven't talked about too much about, sort of transitioning from math to science, right? And what I do now for a living is I use math to understand science. I don't really do math, whereas some of my training was more in math. We don't understand these systems super well. Like a biological dynamical model is typically going to be less detailed and more approximate than a physical one. And so in population biology in particular, we don't get so many bifurcations, but I'm going to come back to that. I'll also say for things like the immune system and systems biology, for understanding how your body works, I think that supports a lot more interesting kinds of bifurcations. But I have done work with bifurcation. Some of it is interesting, and I may try to explain some of it in a minute. But I'm also going to say that right at the very beginning, and what Dr. Fang talked to you about too, R0 equals 1, right? If we're in Kaohsiung and R0 is bigger than 1, and we're worried about dengue, and if we're in Taipei and R0 is less than 1, we're not worried about dengue, that's a bifurcation. And not only is that a bifurcation, we can analyze the sign of that bifurcation, and we get really interesting answers.
It's like pitchfork or something.
So it is transcritical. It's transcritical, and it's a special kind. So when I learned dynamical systems, I was told there's only one transcritical bifurcation. Because up to symmetry, there's only one transcritical bifurcation. But in biology, and in particular, in particular with the R0 bifurcation, the symmetry isn't there because we're looking at disease incidence equals 0. And so it matters what's happening on the positive side, and in some sense it doesn't matter what's happening on the negative side. And so this R0 equals 1 bifurcation is typically what I would call a forward bifurcation, and it's stable in the sense that when R0 is a little bit bigger than 1, we have susceptible depletion, which I know is a topic that you've heard about, that balances it, and so we have a smooth transition. But that's not always the case. There are some diseases for which that bifurcation can go in the other direction, and what we get is a catastrophic change, that we can go just a little bit above R0, we can go epsilon above R0 equals 1, but have a non-tiny amount of disease, and it's also not trivially reversible. So the bifurcation theory that's most relevant is very simple, but it's very interesting, and it's biologically useful. It actually translates. So for example, malaria, there's the question of, I have time. So and there's a question that's really a bifurcation theory question, or the sign of the bifurcation question, which is this. People in highly malarious areas, the children, some of them die, and it's a problem, and we don't like malaria. The adults don't really pay very much attention to malaria. They don't even necessarily know if they have malaria or flu, and they don't seek treatment. And there's a question now that conditions have gotten better, whether the adults not seeking treatment and then becoming long-term spreaders of malaria, whether that's an important part of malaria persistence. That this might be a backward bifurcation, that maybe if we could eliminate malaria for 2 or 3 years in some parts of Africa, maybe it wouldn't come back because of this mechanism of adults feeling sick and seeking care. So that's a case where you have a practical biological question that you can think about using a simple bifurcation theory.
Thank you.
Okay. We thank you. Thank you, Professor. Thank you for your excellent lecture and sharing your highly valuable experience. I think today our students learn a lot from you. You are an excellent role model for all of them.
It's kind of you. I'm sorry again that I had some problem with my equations.
Okay, so I close this section. Thank you all for your all your attention. Thank you. Thank you. Very good.
Oh, good.
Wonderful.